Nasopharynx, oropharynx and laryngopharynx. Extents of all these three parts are important. So pharynx extends vertically from the base of the skull to the esophagus, and it acts as a common passage for food and air. Okay? Food will enter into the pharynx and air will enter into the larynx. Okay, there is a crisscross in between the channel of food and channel of air. Okay, so measurement length is almost 12 to 14 centimeter. Width is about 3.5 centimeter wide in the uppermost part of the pharynx, that is narrow pharynx. In the region of narrow pharynx, it is widest, and 1.5 centimeter, that is narrowest at the pharyngoesophageal junction, where pharynx join with the esophagus. Next is the location, where it is located. It is situated behind the nose, mouth and larynx, and it extends from the base of the skull to the lower border of cricoid cartilage. This level is important. Pharynx ends here at the lower border of cricoid cartilage, that is lower border of C6 vertebra. You can remember extent of larynx is also up to this level, lower border of C6 vertebra. Okay, where it continues as esophagus. Larynx continues as trachea, and this pharynx continues as esophagus. Okay. Now we will discuss the boundaries. Boundaries and external relations of the pharynx. Superiorly, there is base of the skull, posterior part of body of the sphenoid, and basilar part of the occipital bone. Inferiorly, there is esophagus. Posteriorly, there is prevertebral fossa and C1 to C6 cervical vertebra. Anteriorly, there is communication with the nose, mouth and larynx. Tube, thyroid apparatus, carotid arteries, common internal and external, and last four cranial nerves. So these are the relations of the pharynx. Now we will go to the subdivisions. The cavity of the pharynx is divided into three parts as follows. First, nasopharynx. It lies behind the nasal cavity. Oropharynx lies behind the oral cavity, and laryngopharynx lies behind the larynx. So these are the three parts: nasopharynx, oropharynx and laryngopharynx. দাদা হচ্ছে না আবার হ্যাঁ ঠিক আছে Okay, so see here, this is the external nasal opening, nasal cavity, and posterior to the nasal cavity, this bluish colored one, this is nasopharynx. Here is the oral opening, oral cavity, posterior to the oral cavity, here is the oropharynx, and here you can see this is position of the larynx. Posterior to the larynx, the yellow colored one is the laryngopharynx. Okay. So you can see food is coming through the mouth, entering into the entering via the oropharynx, laryngopharynx into esophagus, and air is coming from the nasal cavity and entering into the larynx via this laryngeal inlet. This is schematic diagram: nasal cavity, nasopharynx, oral cavity, oropharynx, that is laryngeal cavity, and here is the laryngopharynx. And this arrow, green arrow, is showing the passage of food, red arrow is showing passage of the air. So there is a crisscross. Okay, so during food intake you cannot breathe, you have to close this laryngeal inlet. Okay, it closes spontaneously. So pharynx is a mucomembranous tube. Length is 12 to 14 centimeter, width 3.5 centimeter in the nasopharynx and 1.5 centimeter at the pharyngoesophageal junction, that is narrowest. Subdivision: narrow nasopharynx behind nasal cavity, oropharynx behind oral cavity and laryngopharynx behind larynx. Okay. Now we will discuss this in individual parts, these three parts individually. First we go to the nasopharynx. Nasopharynx or epipharynx, it is situated behind the nasal cavity and above the soft palate. Boundaries, roof is formed by body of the sphenoid and basilar part of the occipital bone. Floor is formed by soft palate and pharyngeal isthmus. These terms are important. Pharyngeal isthmus is the junction between nasopharynx and oropharynx. Pharyngeal isthmus communication with oropharynx. Anterior wall is formed by posterior nasal aperture. Here is communication with the nasal cavity, and posterior wall is formed by anterior arch of C1 vertebra, that is atlas. Okay. And lateral wall, here you will see medial pterygoid plate of sphenoid, and this roof and posterior wall of the pharynx forms a continuous slope. Okay. See, this is nasopharynx. This roof and posterior wall is forming a continuous slope. What are the important features of nasopharynx? Nasopharynx shows these features: nasopharyngeal or pharyngeal tonsil, adenoid. Okay, adenoid in some children, this adenoid get enlarged and it will cause difficulty in breathing. So, it is a, what is adenoid? It is a median conical mass formed by aggregation of lymphoid tissue beneath the mucous membrane, and it is prominent in children and atrophies in adults. It projects forward from the junction of roof and the posterior wall of the pharynx. Okay. So, this is the location of the adenoid. Next, nasopharyngeal bursa or verticulum, and that extends upward into the substance of nasopharyngeal tonsil from its apex. Okay. So this is pouch and pharyngeal opening of auditory tube is present here. In your sagittal section viscera of head neck, you have seen this opening, pharyngeal opening of auditory tube, that you have to see. Okay, it is important structure. It lies in the lateral wall of the nasopharynx, 1.25 cm behind the inferior nasal concha, and it is a triangular opening. Position is important. It lies about 1.25 cm behind the inferior nasal concha, and other end opens in the middle ear, auditory tube. Okay, or pharyngeal opening of the auditory tube. Then tubal tonsil, the upper and posterior margin of the pharyngeal opening of auditory tube are elevated by aggregation of lymphoid follicles forming the tubal tonsil. Okay. It is located near this opening, tubal tonsil. Then two mucus folds. There are two mucus folds that extend from this tubal tonsil as follows. These two folds are extending towards the tongue, downward. Salpingopharyngeal fold, it passes vertically downward on the lateral wall of the pharynx and contains the salpingopharyngeus muscle. Another fold is salpingopalatine fold. It extends downward and forward to the soft palate and contains the levator valley palatine muscle. Okay, it helps in elevation of the soft palate, levator valley palatine. Now, pharyngeal recess or fossa of Rosenmuller. It is a deep depression behind the tubal elevation. Behind the location of tubal tonsil, there is a deep depression that is known as pharyngeal recess or fossa of Rosenmuller. Okay, so these are the important features you have to see in the nasopharynx. See here. In this section, we have cut open the pharynx from behind. Here, nasopharynx is above, in this part is the oropharynx. See, here is the nasal concha, you can see posterior nasal opening. So, this part will form the nasopharynx. Here is the oral opening, you can see the soft palate. So, here this part is the oropharynx in this part, and this is laryngeal opening. You can see the epiglottis, epiglottic fold. So, this is forming the boundaries of laryngeal inlet, and so behind this inlet will lie this laryngopharynx. See, this arrow is showing extent of these three parts: nasopharynx, oropharynx, laryngopharynx. Okay. And below this level, below the laryngopharynx, it will continue downward as the esophagus. And this part is narrowest at the junction between laryngopharynx and esophagus. Okay, pharyngoesophageal junction. And it is widest in the upper part in this nasopharynx. Schematic diagram. See, this is posterior nasal opening or guana. Here you can see those nasal concha, nasal septum like this. Here is the opening of the auditory tube. On the posterior and upper part of the opening, there is tubal elevation containing tubal tonsil. And in the above, there will be one enlargement that will be at the junction of roof and posterior wall. There will be location of the adenoid. Okay. And posterior to this tubal elevation, in this region will be deep depression, fossa of Rosenmuller. Here will be the oropharynx, oral cavity, posterior opening. Here is the uvula, soft palate and uvula, and this is laryngeal inlet. On the two sides of the inlet, there is important fossa that is known as pyriform fossa. Pyriform fossa boundary is asked very frequently in your Viva, also given in your OSPI. Okay. And also there is some clinical importance of this pyriform fossa. Comes as a short note in your theory. See the different features. Nasopharynx. Okay. This is anterior boundary formed by posterior opening of the nasal cavity. Anterior arch of atlas. Roof is formed by the base of the inferior surface of body of the sphenoid and basic occiput, base is sphenoid. Okay. And floor is formed by the connection between the nasopharynx and oropharynx. And these features you can see, there is the tubal opening, this is the tubal tonsil, here is the location of pharyngeal tonsil or adenoid, and posterior to the tubal tonsil, here is the fossa of Rosenmuller. Okay, 3. Demarcation of 3 is fossa of Rosenmuller. And two folds are extending from this opening of auditory tube. This fold is known as salpingopalatal fold, and here is salpingopharyngeal fold containing the muscles of same name, salpingopharyngeus and salpingopalatine. Okay, salpingopalatine also known as levator veli palatini. Okay, it will hold elevate the soft palate and uvula. Here is the soft palate. You can see up to this bone is extending, so up to this is hard palate. After that, this part is soft palate. It is elevated by this muscle, levator veli palatini, which is located deep to this fold, salpingopalatal fold. Okay, salpingo means this tubal opening is known as salpingo. Okay. So features of nasopharynx: nasopharyngeal tonsil or adenoid, nasopharyngeal bursa. See, within this adenoid, there is a pouch extending deep into its tissue. That is so nasopharyngeal bursa, pharyngeal opening of auditory tube, tubal tonsil, mucosal fold, salpingopharyngeal, salpingopalatine fold, and pharyngeal recess. So these are the. Now, what are the important features of oropharynx? You will see the palatine tonsil, tonsillar fossa, palatoglossal arch, palatopharyngeal arch, liquid tonsil, median and two lateral glossal epiglottic folds and vallecula. All these you will see next. Then laryngopharynx have these three important features: laryngeal inlet, piriform fossa, epiglottic folds. Okay. Okay, so the nasopharynx, similar to the nasal cavity, is lined by pseudostratified ciliated columnar epithelium. These epithelium linings are important for your MCQs. Nasopharynx has ciliated columnar epithelium. Three different parts have three different type of epithelial lining. Okay, and the nasopharynx is the widest and non-collapsible part of the pharynx. Now these are the different clinical aspects. Here you can see this adenoid has been enlarged. This is hypertrophic adenoid. Clinically, the enlarged pharyngeal tonsil is called adenoid. It may obstruct the airway passage at the nasopharynx, and this results in oral breathing, and it may cause spread of infection to the meningeal cavity to the auditory tube, because the opening of auditory tube is lying nearby to this adenoid. So what is the treatment? Adenoidectomy. It is surgical removal of the adenoid. You can see at the junction of roof and posterior wall this enlarged thing. This is hypertrophic adenoid, and here is opening of the eustachian tube. In ENT, you have to locate this opening of the eustachian tube in your patients. Okay. Next, we go to the oropharynx. Oropharynx lies behind the oral cavity, extends from the pharyngeal to the epiglottis below. Boundaries are pharyngeal isthmus through which oropharynx communicates with nasopharynx. Floor is formed by the posterior one-third of the tongue and interval between the tongue and epiglottis. Anteriorly, there is oropharyngeal isthmus through which oropharynx communicates with the oral cavity, and posterior one-third of the tongue. These are forming the anterior boundary. Posterior boundary, bodies of C2 and C3 vertebra. For nasopharynx, you have seen anterior arch of the atlas, C1. For oropharynx, two vertebra, C2 and C3. Okay. And lateral wall, it is formed by the pterygomandibular raphe, mandible, thumb, and hyoid bone. So these are the boundaries of the oropharynx. Now, important features of the oropharynx. Palatine tonsil. There is one palatine tonsil on each side in the lateral wall of the oropharynx. It is located into a tonsillar fossa. You have seen the viscera, sagittal section of head and neck? Yes or no? In your dissection class, you have seen. So all these features you have seen in those viscera. You can see features in the nasopharynx, oropharynx, and laryngopharynx. Okay. So palatine tonsil, there is one palatine tonsil on each side in the lateral wall of oropharynx. It is located into the tonsillar fossa. Tonsillar fossa boundary is important. It is triangular depression in the lateral wall of oropharynx. It is the palatine tonsil. Boundaries of tonsillar fossa: anteriorly palatoglossal arch, posteriorly palatopharyngeal arch, inferiorly posterior one-third of the tongue. Apex is the meeting point of anterior and posterior margins of the soft palate. Palatoglossal arch, it is a mucosal fold that runs downward and forward from the palate to the lateral margin of the tongue, and it contains the palatoglossal muscle. So palatoglossal arch is forming anterior boundary of this tonsillar fossa. Then palatopharyngeal arch, It is mucosal fold that runs downward and backward to the pharyngeal wall. It contains palatopharyngeus muscle. Okay. Then lingual tonsils. These are mucosal aggregation of lymphoid follicles on the dorsum of the posterior one third of the tongue. Okay, so this is lingual tonsils and median and lateral glossoepiglottic folds. They glossoepiglottis, so it will connect tongue and epiglottis. So these names are given according to their location, okay, and the structures they are connecting. Ariepiglottic means it is connecting epiglottis and arytenoid cartilage. So median and lateral glossoepiglottic folds it connect the anterior surface and edges of the epiglottis respectively to the tongue. That's why glossoepiglottic, tongue and epiglottis. Then epiglottic vallecula. You have to look at the vallecula. They are shallow fossa between median and lateral glossoepiglottic folds. Okay. Okay, so this is location of the oropharynx. See, these are the location of tonsil. This structure you can see, this is tonsil, and here is the posterior one third of the tongue where lies the lingual tonsil. Okay. So oropharyngeal isthmus, its boundary is asked in your viva. See, these are the location of tonsil. Here is the posterior one third of tongue containing lingual tonsil. Here is soft palate. On the two sides, the arches, palatoglossal arch, superiorly soft palate, inferiorly dorsal surface of posterior one third of the tongue, and laterally this palatine tonsil is located. So, oropharyngeal isthmus is an arched opening between two palatoglossal folds through which the oral cavity communicates with oropharynx. These are the boundaries: superiorly bounded by soft palate, inferiorly by dorsal surface of posterior one third of the tongue, laterally by the palatoglossal arch on each side. And what are the functions? Oropharyngeal isthmus closes during deglutition to prevent the regurgitation of food from oropharynx to the mouth. Okay. During deglutition, it closes. How it closes? By elevation of this posterior one third of the tongue and approximation of this palatoglossal arch. Okay. Next, we go to laryngopharynx. So laryngopharynx lies behind the laryngeal inlet and it extends from upper border of epiglottis to the lower border of cricoid cartilage. This extent is very important. Extend from upper border of epiglottis to the lower border of cricoid cartilage. Next, what are the boundaries? Superiorly bounded by oropharynx, inferiorly bounded by esophagus, anteriorly there is laryngeal inlet, posterior surface of larynx, posteriorly bodies of C3 to C5 vertebra, laterally there is thyroid cartilage. Now what are the features of the laryngopharynx? Laryngopharynx presents laryngeal inlet, piriform fossa. Laryngeal inlet, inferiorly the laryngopharynx communicates with the laryngeal cavity through this laryngeal inlet. Okay. It is boundary of this laryngeal inlet is very important. This is also asked in your viva. So laryngeal inlet boundary, it is bounded superiorly by the upper margin of epiglottis, below and behind by the mucous fold, and on each side by the ariepiglottic fold. So these are the boundaries of the laryngeal inlet. Another important feature is piriform fossa. We will discuss in details piriform fossa. See here, this is the laryngeal inlet. On the two sides of inlet, these depressions are known as piriform fossa. In the boundaries of laryngeal inlet and anteriorly, there is apex of the epiglottis. Posteriorly, there is arytenoid cartilage. On the two sides, ariepiglottic folds. Okay, these ariepiglottic folds are connecting they are containing two cartilages of larynx, cuneiform and corniculate. These are small beads-like cartilages, okay, embedded within this mucous pore. Okay, understood? So this is the boundary of laryngeal inlet, and posterior to the inlet will lie the laryngopharynx. Here also you can see this is laryngeal inlet. Epiglottis is forming anterior boundary. On the two sides, periepiglottic folds, posteriorly the arytenoid. And here is the location of piriform fossa on the two sides of the inlet. So the piriform fossa is very important. Comes as a short note. It is also called piriform sinus or smuggler's fossa. Okay. It is a depression in the lateral wall of the laryngopharynx, one on each side of the laryngeal inlet. Here is the location of piriform fossa. This is laryngeal inlet. On each side of the laryngeal inlet, the fossa is located. What are the boundaries? Medially bounded by aryepiglottic fold. Here is aryepiglottic fold forming the medial boundary. Mucosa covering the medial surface of lamina of the thyroid cartilage and the thyroid membrane. Cartilage. So lateral boundary is mucosa covering the medial surface of lamina of thyroid cartilage. All the terms are important. All the whole of the line you have to say if you are asked in your viva. Above there is lateral glossoepiglottic fold. Here will be the lateral glossoepiglottic fold connecting tongue with the epiglottis. Separate the piriform fossa from vallecula. These are the two vallecula lying between median and lateral glossoepiglottic fold. Location of vallecula and lateral to the lateral glossoepiglottic fold. Glossoepiglottic fold and aryepiglottic fold. Understood? Because the boundary combination internal laryngeal nerve, superior laryngeal vessels pierce the thyroid membrane and transverse underneath the mucous membrane of the floor of the piriform fossa. See, the superior laryngeal vessels have been shown this dotted structure. Okay, this will enter through this thyroid membrane, pierce this thyroid membrane and lie in the floor of the piriform fossa. This piriform fossa is deep in ruminating animals. Okay, when in cows, buffaloes, they can accumulate food in this piriform fossa, and then later on they can ruminate those food. They will regurgitate back into the mouth and ruminate. Now, what is the clinical importance of piriform fossa in human? It is also known as smuggler's fossa, because why it is known as smuggler's fossa? It is important for your viva. Sometimes the piriform fossa is artificially deepened by smugglers and used to hide diamonds. Okay, to export diamonds from different countries. A perforation is very common in this region. Piriform fossa may get perforated during endoscopy. Okay, and foreign bodies are also damage to the internal laryngeal nerve. Accidentally ingested foreign bodies may get lodged in this piriform fossa. Very frequent location of lodgement of foreign body. Okay, like fish bone or other foreign bodies, sharp foreign bodies. So during their removal, the internal laryngeal nerve may get damaged. Okay, so there will be difficulty during voice production if internal laryngeal nerve is damaged. Now this is histology of pharynx, structures in the wall of the pharynx. Wall of the pharynx consists of this following layer consisting of what? First is mucosa. Innermost is the mucous membrane. Then there is some mucosa. Okay. After that there is pharyngobasilar fossa. Pharyngobasilar fossa. Which one is the pharyngobasilar fossa or pharyngeal aponeurosis? Then there is muscle cord. The solid red colored one is the muscle cord. And then there is buccopharyngeal fossa or loose areola cyst. Buccopharyngeal fossa. These are the layers of the pharynx. Here this is a longitudinal section. Here also you can see the pink one is the mucous membrane. Then there is some mucosa, then pharyngobasilar fossa, then muscle layer. Here in the pharynx, the muscle layers contains three circular muscles. They are cone shaped and one cone is inserted into other. Okay, so from above downward, the muscles are superior, middle, and inferior constrictor muscles of the pharynx. Then outside it will be the buccopharyngeal fossa, consists of loose areolar connective tissue. Okay, so these are the layers of the pharynx. This is also asked in your viva. Okay, mucous membrane of pharynx, it contains considerable amount of elastic tissue and the pharynx is lined by non-keratinized stratified squamous epithelium except in the nasopharynx. Rest of the part, oropharynx and laryngopharynx is lined by non-keratinized stratified squamous epithelium and nasopharynx is lined by respiratory epithelium lined by pseudostratified ciliated columnar epithelium. Now tonsils, this sub-epithelial collection of lymphoid follicles produce Waldear's ring. Waldear's ring is important. It comes as a short note. Okay, Waldear's ring boundary. So Waldear's ring that has pharyngeal tonsil, tuval tonsil, palatine tonsil, and lingual tonsil. Some mucosa in the upper part of pharynx it condenses to form the pharyngobasilar fossa. Pharyngobasilar fossa or pharyngeal aponeurosis, it is a fibrous thickening of the pharyngeal submucosa. It lines the pharyngeal muscles internally and it is thicker near the base of the skull where it fills the gap between the skull and upper border of the superior constrictor muscles. And this gap is called sinus of Morgagni. Posteriorly, the pharyngobasilar fossa forms the pharyngeal raphe. Now, muscle cord. The pharynx consists of two layers of striated muscle, outer layer of three pairs of constrictor muscle, and inner layer of three pairs of longitudinal muscles. So, these are forming the muscle cord. So, these are the constrictor muscles. Muscles of the pharynx consist of striated muscle arranged in two layers, outer circular layer, three pairs of constrictor muscle, superior, middle, inferior constrictor, inner longitudinal layer consists of three pairs of muscle, allopharyngeus, palatopharyngeus, salpingopharyngeus. These are forming the longitudinal layer. Now we will discuss the constrictor muscles. Constrictors of pharynx form the lateral and posterior wall of the pharynx, originate anteriorly and pass anteromedially to get inserted on the midline fibrous pharyngeal raphe. Okay, pharyngeal raphe above it is attached to the basic occiput. Okay, you have seen the attachment in your occipital bone. And these constrictors are arranged in a specific manner so that the lower fibers of superior constrictor are overlapped by upper fibers of middle constrictor and lower fibers of middle constrictor are overlapped by upper fibers of inferior constrictor. You can see here cone shaped. This is a longitudinal section. So you can see the overlapping. Lower part of superior constrictor overlapped by middle constrictor. Lower part of middle constrictor overlapped by the inferior constrictor. Okay. So there are superior constrictor, between superior middle constrictor, between middle and inferior constrictor, and below the inferior constrictor. Embryo Physiologically, this constrictor of pharynx develop from fourth and sixth pharyngeal arches and hence supplied by cranial part of accessory nerve through the vagus. Nerve supply is important for MCQ. Absence of constrictor, there will be deglutation. Nerve supply supplied by fiber from cranial part of accessory through pharyngeal branch of vagus. Thyropharyngeus is supplied by pharyngeal plexus and external laryngeal branch of vagus nerve, and cricopharyngeus is supplied by recurrent laryngeal branch of vagus nerve. So these are exceptions of this nerve supply. Rest of the part is supplied by cranial part of accessory nerve, and these two muscles supplied by other. Thyropharyngeus from pharyngeal plexus and external laryngeal branch of vagus. Cricopharyngeus from recurrent laryngeal branch of vagus. Okay. See the location. Here constrictor. This is constrictor. Inferior constrictor. And see the gaps. Gaps are present above the superior constrictor and between the base of the skull and superior constrictor. This is known as sinus of Morgagni. These gaps are important because important structures are passing through these gaps. Okay. So above the superior constrictor, here is location of sinus of Morgagni. In between superior and middle constrictor, second gap. In between middle and inferior constrictor, third gap. And below the inferior constrictor will be the fourth gap. And all these constrictor muscles here in the midline posteriorly, they meet in the pharyngeal raphe. Okay. This is important schematic diagram. Here is the pharyngeal raphe, superior, middle, inferior constrictor, sinus of Morgagni, and all those gaps. Okay, so these are the origin and insertion. If it comes as a short note, you have to write also origin insertion of individual constrictor muscles like this. Okay, so origin is superior constrictor, pterygoid hamillus, pterygomandibular raphe, mandible at the posterior end of mylohyoid line, side of tongue. Insertion into the pharyngeal tubercle, median raphe. All those insertion is median raphe. You remember it like this. Middle constrictor origins are different. Middle constrictor lower origin from lower part of stylohyoid ligament, lesser cord to upper border of greater condylohyoid. Inferior constrictor has two parts. This is asked sometimes to name the two parts of this inferior constrictor. Thyropharyngeus, cricopharyngeus. Thyropharyngeus originate from oblique line of lamina of thyroid cartilage. Cricopharyngeus from the cricoid cartilage, and nerve supply is cranial root of accessory nerve. So this chart is showing the origin insertion of this constrictor muscle. Okay. Now we will discuss the gaps between these constrictor muscles and what are the structures passing through this gap. So there are gaps in the constrictor muscles. First gap is gap between the base of the skull and upper border of superior constrictor. This is called sinus of Morgagni. Structure passing through this gap is auditory tube, ascending palatine artery, and palatine branch of ascending phalanx between base of the skull and this superior constrictor. So these are the structures passing auditory tube. Here is the levator valley palatini. This is ascending palatine artery. Here is the palatine branch of ascending pharyngeal artery. Okay, so this is asked in your viva. What are the structures passing through sinus of Morgagni? Now, gap between the superior and middle constrictor. Superior and middle constrictor. Through it passes thyropharyngeus muscle and pharyngeal nerve. Thyroid membrane enter into the laryngeal cavity to supply the larynx. Okay. Next, between the lower border of inferior constrictor and esophagus, inferior laryngeal vessel. Here, recurrent laryngeal nerve and inferior laryngeal vessel. Here, remember, within the larynx. In the anal canal, there is a watershed area, pectinate line. In larynx also there is a watershed area. Okay, watershed area is located near the vocal cord. That will be discussed during larynx. So above the vocal cord, supplied by the superior laryngeal vessels and the internal laryngeal nerve, and below the vocal cord, it is supplied by this recurrent laryngeal nerve and inferior laryngeal vessels. Okay, that's why this part is known as watershed line of the larynx. Jenker's diverticulum or Killian's dehiscence. This is also comes as a short note. What is it? Pharyngeal or pharyngeal mucosa. This is inferior postcentral muscle. It has two parts. You are seeing this upper part is hypopharyngeus. This is obliquely placed. And it is transversely placed. This is picopharyngeus coming from the picot cartilage. In between these two parts, in this area, here the mucous membrane is out sometimes and forms this Jenker's diverticulum, or and this gap in between this thyropharyngeus and picopharyngeus, this gap is known as Killian's dehiscence. Dehiscence means weak area. Weak area in the pharynx, and through this weak area, mucous membrane pouches out as Jenker's diverticulum. Okay, understood? So, Killian's dehiscence is weak posterior part of the pharyngeal wall that lies between picopharyngeus and thyropharyngeus part of the inferior constrictor of pharynx. And in case of the excessive pressure within the lower part of pharynx, the mucosa of the pharyngeal wall fills out to the Killian's dehiscence to form the Jenker's diverticulum. Okay, it is named after Gustav Killian, German laryngologist, and it may result due to neuromuscular of the picopharyngeus muscle. This is the etiology. It occurs because the two parts of the inferior constrictor have different nerve supplies, and the propulsive thyropharyngeus is pharyngeal and sweet supplied by recurrent laryngeal nerve. And in neuromuscular incoordination, if the picopharyngeus fails to relax when the thyropharyngeus contracts, this upper one will contract, lower one will relax. Okay, because of different nerve supply. And this will help in propulsion of food towards the esophagus. So, if this due to this incoordination, it fails to occur, then the bolus of food is pushed backward and tends to produce a diverticulum. That is known as Jenker's diverticulum. The signs and symptoms, it may be asymptomatic, or there will be difficulty in swallowing, dysphagia. Okay, all the food will accumulate here and no food will go down. There will be dysphagia. So, Jenker's diverticulum definition, also known as pharyngeal pouch or diverticulum of pharyngeal mucosa. Killian's dehiscence is weak part of posterior pharyngeal wall lying between picopharyngeus, thyropharyngeus. Formation pharyngeal mucosa through Killian dehiscence. Formation of Jenker's diverticulum. So, cause is neuromuscular incoordination. Signs, symptoms, it may be asymptomatic, or there may be dysphagia and regurgitation of food and cough. Okay, these are the symptoms and signs. And diagnosis, you can see this pouch in barium solo X-ray of the esophagus, barium solo radiography, and also can be diagnosed by endoscopy. Okay, understood? So, Jenker's diverticulum you prepare as a short note. These are the longitudinal muscles of pharynx, 3 pairs. Here you can see coming from, all are located in the posterior border of lamina of thyroid cartilage. One is coming from styloid process, that is known as stylopharyngeus. One is coming from the or opening of auditory tube, that is salpingopharyngeus. One is coming from soft palate, that is palatopharyngeus. So these three are forming the longitudinal muscles of pharynx. Thyroid cartilage. See, the same like thyroid cartilage, all are inserted into posterior border of thyroid cartilage, and these muscles elevate the larynx and shorten the pharynx during swallowing. So this is function of this longitudinal layer. Understood? Origin and insertion of these three muscles, they elevate the origin styloid process, palatopharyngeus, upper surface of palatine aponeurosis, salpingopharyngeus, lower part of cartilage of auditory tube, insertion into posterior border of lamina of thyroid cartilage. Stylopharyngeus is supplied by glossopharyngeal nerve, palatopharyngeus and salpingopharyngeus by cranial root of 11th cranial nerve, that is spinal accessory nerve. This chart is showing the origin, insertion, nerve supply of this longitudinal muscle. This is not muscles of mastication. Muscles of mastication different. Okay. Okay, so the nerve supply, blood supply, and lymphatic drainage of pharynx. Nerve supply is by pharyngeal plexus formed by pharyngeal branch of vagus carrying fibers of cranial accessory nerve, pharyngeal branch of glossopharyngeal nerve, and pharyngeal branch of superior cervical sympathetic ganglia. Motor supply is by cranial root of accessory nerve through vagus nerve, except stylopharyngeus and glossopharyngeus. Inferior constrictor receive additional supply from recurrent laryngeus. Sensory by this nerves, glossopharyngeal, vagus, pterygopalatine ganglion, maxillary nerve, and lesser palatine nerves. Taste sensation from vallecula and epiglottis is carried by internal laryngeal branch of vagus. And secretomotor supply is coming from parasympathetic fibers from lesser palatine branches of pterygopalatine ganglion are secretomotor for the mucous glands of the pharynx. Arterial supply by these following arteries: ascending pharyngeal branch of external carotid, ascending palatine tonsillar branch of facial artery, dorsal lingual branch of lingual artery, and greater palatine and pharyngeal branch of maxillary artery. Venous drainage is by pharyngeal venous plexus, drains the venous blood from pharynx situated in posterolateral aspect of pharynx over the middle constrictor, and this drains into internal jugular or facial vein. Lymphatic drainage drains into retropharyngeal and deep cervical nodes. So these are the nerve supply, arterial blood supply, and lymphatic drainage. Deglutition, it generally does not come, but you just know the process. Three stages are there. First is the food bolus is located in the mouth. It can be controlled by voluntary contraction of different muscles. Second stage, bolus is lying in the pharynx. It is involuntary. Third stage is also involuntary. Okay, read it out. So what are the clinical aspects of this deglutition? Dysphagia, it is difficulty swallowing, causes of carcinoma of the esophagus, cardiospasm or cardiac achalasia. Okay, what is cardiac achalasia? It is failure of lower esophageal sphincter due to congenital absence of ganglion cells in mesenteric plexus of esophageal muscle, and it causes accumulation of food in the esophagus, that is cardiac achalasia cardia. This achalasia cardia comes and short note or important clinical aspect, part of a long question. What is achalasia cardia? Failure of lower esophageal sphincter due to congenital absence of ganglion cells in mesenteric plexus of esophageal muscle. It causes accumulation of food in esophagus. And dysphagia can occur due to carcinoma esophagus and cardiospasm. Okay, pharyngeal space is located around constriction of pharynx. One is retropharyngeal space and two lateral pharyngeal or parapharyngeal space. These spaces are important because during infection, they are location of pus and you have to drain the pus from these spaces. This abscess, retropharyngeal abscess, parapharyngeal abscess, you have to read during ENT. Okay, okay, that's all. Any queries? Okay, so prepare Killian's medicine and auditory tube, all those I have told. Pyriform fossa to prepare for your short note, okay? And different boundaries and important features, presenting parts and important features are important for your vulva and viscera. And prepare the boundary of Waldear's ring above adenoid on the tubal tonsil, palatine tonsil, lingual tonsil. So you prepare pyriform fossa, Waldear's ring, and Killian's striations as short note important. Okay, that's all. Thank you.
Transcription is what? We have DNA which have double helix structure and there is base pairing and we have RNA templates. The basic difference between that is only that is present of uracil against thymine. Understand? In DNA, thymine is present. In RNA, uracil is present against thymine. Okay? So you will say this is same like a process of replication. I think you all of you know the replications also. Replication is what? Copy of DNA. Formation of copy of DNA with the help of DNA polymerase enzyme. Okay? So same also in this process, what happened? There is also a formation of RNA template with the help of enzymes, with the help of factors and some other units. Okay? We can differentiate this transcription in prokaryotes and eukaryotes. There is the process of transcription in eukaryote is differ. Why it is differ? Because these RNA molecules are formed in nucleus which is not well present in prokaryotes. And after the formation of these RNA templates, these become going for some post-transcriptional modification. Then it is going for further process called translation in the cytoplasm. So these have some differences. Okay? Before that, we understand this topic very well. Okay? So first is definition. What is the definition of transcription? Transcription is formation of RNA molecule by using DNA as a template, as a modulate molecule. As a modulate molecules. You already know that DNA having template strand, antisense strand, sense strand. What is the meaning of that? You already know. Okay? That result the transfer of the information stored in double-stranded DNA into a single-stranded RNA, which is used by the cells to direct the synthesis of protein. Okay? Now you simply understand what the central dogma says. This is being our DNA molecule which forms single strand RNA molecules and it further going for next process and expressed in the form of protein. Expressed in the form of protein. And this is called formation of RNA molecule by use of DNA template in small fragments. what is different between replication and transcription? The replication use whole DNA strand, but in transcription they used only a small fragment of DNA molecules. Okay, from this DNA molecule it forms these RNA molecules. This is called transcription. This is called transcription. So what is the definition of transcription? That is synthesis of RNA molecule by using of DNA as a template, transfer of this information, because you already know DNA RNA is a genetic material and it is contains some genetic code which is responsible for different expressions of proteins in the body. Okay, so by using this DNA as a template, the result in the transfer of information is stored in double-stranded DNA into single-stranded RNA, which is used by the cell to direct synthesis of proteins. Okay, synthesis of protein. Now we can differentiate these transcription in two ways: transcription in prokaryotes, transcription in eukaryotes. Okay, so firstly we discuss transcription in prokaryotes. Now we understand all these topics in very well in the board. So how this question, if the question is asking in your examination, it is asked in long answer questions, it is asked in short answer questions also. The question is asking what is transcription, first you give the expression of protein inside the cell. It is called transcription. Okay, so this is the definition. Here we require polymerase enzyme. Okay, ribonucleic polymerase enzyme. We discuss bacterial transcription here. What we discuss? Bacterial transcription here. Okay, it is a transcription of prokaryotes, and what is the requirement? Requirement for this transcription, requirement discuss one by one, and it is a very easy process. And there nucleotide triphosphate. Nucleotide triphosphate. What is basically what we do here? What we do here? In the DNA molecule, there is the presence of thymine. In the RNA molecule, there is the presence of uracil. Okay? So this is basic difference. This transcription, there are mainly three steps. The first one is initiation. Second one is elongation, and the third one is termination. Okay, this being the three steps. So if the question is asking write about the transcription of prokaryotes, you will write in this format. First you describe the transcription, then you will give the RNA-PAs. RNA-PAs is an enzyme in prokaryotes. There are basically difference between prokaryotes and eukaryotes. In eukaryotes, there are three basic classification of this RNA polymerase enzyme. RNA polymerase enzyme. Which one is that? RNA polymerase first, RNA polymerase second, and RNA polymerase third in eukaryote. We discuss in eukaryote. But in prokaryotes, these are present in combined form. These are present in coordinate molecules forms. So firstly, the RNA enzyme. RNA what? Can you tell me what the meaning of holo enzyme? The enzyme present with core molecules and some prosthetic groups. Okay. So holo enzyme, in the holo enzyme molecules, what is there? It contains enzyme. That means it contains alpha 1, beta, omega unit with a protein called sigma factor. Sigma protein. With a protein called sigma protein. Okay. How it is present? We call this is core enzyme, and with this holo enzyme. We talk about RNAPase, RNA polymerase enzyme in prokaryotes. How it is present? It is present in alpha unit, alpha 1 alpha dash, beta 1 beta dash. This is alpha 1 alpha dash, beta 1 beta dash, and there is a present of omega. Okay, and here there is sigma proteins are present. So now what is the function of this particular separation of these enzymes? These sigma proteins scan the DNA template. Why this protein is scanning this DNA template? It is simply search a appropriate space in the DNA template for the attachment of RNA polymerase enzyme. Understand? Just like that, you are just walking in the ground, and when you want to take the lecture, you are coming in the lecture hall. So what we do? You will scanning the lecture hall to attend the lecture. So to initiate the transcription process, these sigma proteins scanning the DNA molecules and attach at an appropriate space to start transcription. You just easily understand it is scan the DNA molecules. What is this scan? What is it scan? It scans some particular sequences, some base pairing sequences. We call these sequences as Prevno box. You already listened the name. What is the meaning of Prevno box? That is Tata box. Okay, we understand all these things. Okay, what is the function of polymerization? Attachment of nucleotide triphosphate molecules with fresh DNA RNA templates. Okay, it do polymerization. And this alpha dash is used for opening of coil of DNA. How DNA molecules are present? If you read the molecular biology, you easily understand. It is present bind with histone proteins. Okay. So what is that? They firstly open this coiling. Okay. So these are the unit, and all the subunits have different function. Understand? RNAPase In the prokaryotes, they are present in combination form. We call this beta subunit, alpha subunit, omega subunit with sigma proteins. And all these particular subunit have different functions. Okay? So this is called RNPS. Now we understand the template, promoters and NTP regions. Template. Template means what? Simply and this is now you understand the basic thing about that. We have two strands. The first one is template strand. I will ask the question to anyone. Be careful in my class. So this is called template strand. This is called template strand. You understand the terms. First, you understand the terminology of this particular process. And okay, template strand means what? What is the meaning of template strand? It is called self-strand. Negative. Okay. Now from the numbering of this base pair, we start our transcription process. So this is the template. What we required here? Template. We required here this template for the attachment of our RNA polymerase enzyme. Okay. Now we go understand all these things. Now what we required here? Nucleotide triphosphate, that is ATP, ATP, three types of nucleotide triphosphates are required. These three types of nucleotide triphosphates are required because here what we form? We form RNA molecule. That's why uridyl is present against thymine. Uracil triphosphate. Okay. Now promoter regions. What is the meaning of these promoter regions? You understand the terms. You basically now we understand the term that +1. This is +1 position and we call this transcription starting site. Okay. From +1, if we go for this, negative sign upstream. And this is the template which is used for the formation of RNA. All this is stream. Negative sign, positive sign. Okay. Now from this one side, if we are going for next minus 10 position, then there is some sequence of amino acid is present, and we call these sequences TAAT. This is called Tata box. What we call Tata box, and it is called promoter region. What we call this? Promoter region. And then for next is TT. Sequence. Have you understand up to this? What we are here? We just separate the DNA strand, double helix strand, and what is present in the DNA? There is base pairs are present. Okay? From one strand, we start the transcription side from +1. Then it having some sequences, we call this Tata promoter region. This is promoter region, this again is promoter region. So I already tell you what is the function of sigma protease. It scans the promoter region for the attachment of RNA polymerase enzyme. And we called this all these sequences, we called this as transcription unit. We called transcription unit. Understand? This is transcription unit. And the RNA which is formed after the pre-initiation complex, we understand all these things in the next step. So you understand that we have template, promoter regions, and nucleotides. We have some sequences. We call this sequence as promoter region second. Firstly, the sigma factor, sigma proteins scans the DNA molecule and it is attached with these promoter region. After then, it attached with Tata box. There is the attachment of RNA polymerase enzyme. Okay? So this is called transcription unit. I think you understand all these things. Now we discuss one by one all the steps. Initiation, elongation, and termination. We understand all the steps. And okay, we discuss after. There is first step called close complex. Okay. Now the second step is open complex. Open complex. Now the third step is synthesis. Synthesis of nucleotide base pairs. 5 to 10 nucleotides base pairs. Okay, and lastly in the and clearance of promoter sequences. Clearance of promoter sequences. Now we understand all these terms very. So this is the first. It is 3 prime to 5 prime. 3 prime to 5 prime. And we have +1 transcription starting sites. Okay, transcription starting site. After then -10 base pair. What is present? What is present? Very good. Tata box. And again, there is some sequences are present. Please tell me the base pair of these sequences. Tell me base pair. T, TG, TG, T. Now, what is mainly here? What is the main? Why we called? Firstly, the sigma factor which is present in the RNA polymerase enzyme in prokaryote, it scans the Tata box, and here it is attached with that, and it is called closed complex. Or another name of this closed complex is pre-initiation complex. Pre-initiation complex. Okay? Pre-initiation complex. Now, in the next step, what is happening here? There is the formation of open complex. So, open complex means what? There is bubble formation. What is formation here? Bubble formation. You see, in the next step, there is prime, and there is attachment of RNA polymerase enzyme, and here this is formation of bubbles, means it distorted the coiling of the DNA molecules and it forms bubble formations. Okay, so this condition is called open complex. Okay, now after then synthesis of 5 to 10 base pairs. Now what is happening here? There is a function of RNA polymerase enzyme. What is the meaning of polymerization? What is the meaning of polymerization? Attachment of nucleotide base pairs with 3' OH group of these newly synthesized RNA molecules. Newly synthesized RNA molecules. So what is there? 5 to 10 nucleotide base pair. 5 to 10 nucleotide base pair, or addition of 5 to 10 base pair for newly synthesized this RNA template. This being step third. How it is possible? How it is possible? Anyone can tell how it is possible addition of these base pairs from this newly synthesized template with the help of enzyme RNA polymerase. And what it contains in the RNA polymerase? It contains beta subunits. Beta subunits are responsible for polymerization of these nucleotide base pairs. Understand? Nucleotide base pairs. Okay? And after then, clearance of these promoter sequences. What is the meaning of clearance of these promoter sequences? Now these RNA polymerase enzyme change their position in the studied prime direction. Go to 3' direction, and due to this, they can change the position from these promoter sequences, and we called this as clearance of promoter sequences by RNA polymerase enzyme. Clearance of promoter sequences by RNA polymerase enzyme. Now repeat again all these initiation steps. Okay, you listen carefully. If you have any queries, you will ask. We have DNA templates. In the DNA templates, we have template and coding strands. Okay, in the template up to +1, there is transcription starting sites, and we going for upstream direction up to -10 base pairs. There is a presence of Tata box, and we call this Tata box as a promoter sequence. What we call promoter sequence. And if we going forward to same direction in -35 base pair, there is the presence of consequences sequences. What is the need of this promoter sequences, consequences sequences? It will helpful for the attachment of RNA polymerase to this DNA template. How it is attached? With the help of sigma protein. Sigma protein is scanning these DNA molecules. Understand? This sigma protein is scanning these DNA molecules, and it is firstly attached in consequences sequences, then it is attached with particular promoter sequences, which we call Prevno box or Tata box. The another name of this promoter sequences is called Prevno box. Sometimes question is asking, write about Prevno box. You are not confusing here. Prevno box is simply first promoter sequences. Now, in this step, in initiation step, there is first is formation of closed complex. Closed complex means what? Simply the RNA is attached, RNA polymerase is attached with the DNA fragments. RNA polymerase is attached with the DNA fragments. You easily see the diagram if you have any confusion. Simply you see these RNA polymerase enzyme recognize the promoter sequences and attach with that. Binding of RNA polymerase enzyme, and we called this step as a closed complex. What we call closed complex. Now, after the attachment of this RNA polymerase, these can unbind these DNA molecules and formation of bubble. You will see there is a formation of bubble, and we call as open complex. Open complex. And in this initiation transcription complex, there is now in the next step, there is elongation. There is elongation. Up to this, you all understand the meaning of initiation. Now we go for elongation. Don't talk. Trend. Okay. So in initiation, what here there is formation of 5 to 10 base pairs. Now in the next step, there is stretch of this newly formed RNA template by attachment of nucleotide base pairs. Okay. So this is called elongation. This is called elongation. Okay. Elongation process is very simple. You easily see there is nothing to worry. There is stretch of this RNA template, and there is elongation process occur. Elongation process occur. Okay. Now the last one is termination. Termination process in prokaryotes. In prokaryotes, it is to be simple. It is very simple. Okay. Okay. Now there is a spatial base pairing sequences which we call AAU, AA, and CA sequences are present. What is happening here? There is some, in the last step, there is cleavage factors are there. Some cleavage factor and some cleavage and polyadenylation. Polyadenylation complex. Polyadenylation complex. Polyadenylation complex and some proteins which having endonuclease activity. Endonuclease, the particular fragment internally. By this endonuclease activity, it can rebound these newly formed DNA from DNA. It rebound or attach RNA complex. DNA RNA complex from this bubble, and we get separate RNA strand. Separate RNA strand. It is simply called transcription. With the help of some cleavage factors, some polyadenylation sequences, and some these endonuclease proteins, they will cut these fragments and separate out this RNA template and DNA template, and here our motto is over. So what is our motto here? Formation of newly RNA templates. So you will see there is initiation, polymerase terminate, and release RNA. So after then, what is here? There is all the enzymes are separate out, and the RNA fragment which is newly formed is separate out from this DNA molecule. Okay? Understand? Understand the transcription in prokaryotes? I revise transcription in prokaryotes. What is there? There is simply template strand, promoter sequences, consequences sequences. Okay? So what does they do? They simply, there is attachment of RNA polymerase enzyme. With the attachment of RNA polymerase enzymes, it going for three-stage process called initiation, elongation, and termination. In initiation, there is formation of closed complex, open complex, synthesis of 5 to 10 nucleotide base pairs, and lastly, clearance of promoter sequences. In the elongation, stretch of this RNA template, stretching of this RNA template by synthesizing, by addition of nucleotide base pair at 3' OH group of this template, by polymerization, and lastly by some cleavage factor it is removed out. But these are separate in transcription in eukaryotes. Now we discuss transcription in mammalian cells or eukaryotes. Okay? So simply if the question is asking right about the transcription in eukaryotes, you also write it in this form. You also write it in this form. You will give the definition is same for eukaryotes and prokaryotes. Okay? And after that, there is again RNP enzyme, RNA polymerase enzyme. There is RNA polymerase second and third. These having separate RNA polymerase in eukaryotes, in mammalian cells. Okay? And there is a requirement. There is a RNAPase, some factors, and after then, same step. How many steps are there? Speak loudly. 3 steps. Which one? Initiation, elongation, and third one is initiation, elongation, and termination. Okay? And we have 3 different enzymes: RNP first, RNP second, and RNP fourth. So this and helpful for the transcription RNA, because RNA is having 3 types. Okay? So what is there? There is formation of first enzyme. Okay? Polymerase first enzyme. Okay? You learn these things. आपको याद होना चाहिए. There is formation of mRNA. And RNA and RNA. all are the RNA polymerase first, second and third, which is used in the process of transcription in eukaryotes or mammalian cells. Okay. Now the factors are what? These are some special class of proteins. These are some special class of proteins. One that is called, we divided these proteins as firstly as activator, secondly basal factors, and the third one is co-regulator. Third one is co-regulator. We divide these factors: basal second factors, co-regulators and activators. Okay. Now we understand the example, that is the example of activators are second T, second B, second E, and lastly second H. All these are what? All these are basal transcription factors. Basal transcription factors. And lastly co-regulators. And co-regulators are basically formed by transcription factor second D and having Tata binding proteins plus Tata binding proteins and 14 associated transcription factors. 14 associated transcription factors. Okay. So all these are the requirements for transcription process in the eukaryotic cell. In eukaryotic cell. Understand? Eukaryotic cell. Now we understand simply the step of this reaction. Step of this process. Step of this process. It is, if you make a diagram very with proper labeling. अभी एग्जामिनर क्या कर रहेगा? एग्जामिनर जो है उसको इतनी कॉपी चेक करनी है, 150 आपकी कॉपियां हैं, इतने क्वेश्चंस हैं. अभी आपकी कॉपी चेक करने बैठेगा, आप जनरल लैंग्वेज में पैराग्राफ में आप लिखेंगे तो क्या ही नंबर देगा आपको. अगर आप डायग्रामेटिक प्रेजेंटेशन देंगे, कुछ सब हेडिंग्स को बोल्ड लेटर में लिखेंगे और वो कॉपी के सामने इम्फेसिस होके वो चीज आएगी, तो आपको इजीली वो नंबर मिलेंगे. मिलेंगे कि नहीं मिलेंगे? यू विल गॉट इजीली नंबर. इफ योर प्रेजेंटेशन इज गुड, योर मार्किंग इज गुड. इफ योर प्रेजेंटेशन इज वर्स्ट, देयर इज नथिंग टू इम्प्रूव. कोई आपकी कॉपी सुधार नहीं आएगा. लिखना आपको है, किस तरीके से लिखना है. बेटर डायग्राम होना चाहिए, बेटर प्रेजेंटेशन होना चाहिए. ठीक है. नाउ देयर इज व्हाट? देयर इज on this is called transcription starting site. Okay, and if we are going for upstream, there is a presence of and if going there is a GC box. You understand this diagram? Why I am calling upstream, which one calling downstream? Upstream means moving towards direction from starting point. From starting point. Okay? In eukaryotic cell, there is the presence of these sequences which we call Tata box, cat box at particular region. Okay? So there is firstly the initiation, there is the form, and we call this unit as transcription unit. I already tell you in transcription unit. So in this step, there is again simple the step of initiation in prokaryotes and eukaryotes are same. Which one is the step? The first step is formation of closed complex, then open complex, then synthesis of 5 to 10 nucleotide base pair and clearance of premotor region. Okay. Now here there is something different. Different means was Tata binding proteins are there which recognizes Tata box. It is recognized by Tata binding protein, and with that some these transcription factors which attachment of polymerase enzyme with polymerase enzyme, then which we call helicase enzyme. So what is the function of this helicase enzyme? What is the function of this helicase enzyme? Unbinding of DNA coiling structure. Which enzyme is doing this unbinding of these DNA molecules? In DNA replication, this is topoisomerase. Topoisomerase. Okay. So all are associated with that. We understand one by one. We understand one by one. So firstly, there is the attachment protein, and this transcription factor D is what they do. They can bind this DNA fragment. Bind this DNA fragment. Students understand this. It binds the DNA. So firstly, this is called Tata box. So firstly, there is attachment of Tata binding protein. So this is your and after then what I tell you, there is attachment of transcription factor second D. Transcription factor and again there is the attachment of transcription factor A. Transcription factor F. This is transcription factor F here. Okay. This is called initiation complex. We call this as close complex. We call this as close complex. What is happening here? Firstly, there is attachment of Tata binding protein in Tata boss sequences. After then, there is attachment of transcription factor D, which binds the DNA molecules. After then, there is attachment of transcription factor A and transcription factor V with transcription factor F with polymerase enzyme. With polymerase enzyme. Okay? So this is called close complex. This is called close. Now in the next step, open complexes. It is same as prokaryotes. Formation of open complexes. Here polymerase enzyme is there with transcription factor F. Okay. There is retachment of this nucleotide base pair. Okay. Nucleotide base pairs. Nucleotide base pairs. Okay. So this is called formation of pre-initiation complex. Formation of pre-initiation complex, and we will go for in the next step. Don't talk. आवाज नहीं आनी चाहिए. I think time is over. So on Monday we discuss step of this transcription of eukaryotes, elongation process, and lastly termination, because both the processes are very important and it is separate from prokaryotes. Okay, because your time is next आपका DH का क्लास होगा, प्रैक्टिकल्स का क्लास होगा. ठीक है, मंडे में हम डिस्कस करेंगे ये दोनों स्टेप and after then we discuss post-transcription modifications with their inhibitors. Okay, in detail. I think all of you understand this. Okay, we will also repeat transcription of eukaryotes on Monday. So all of you read this topic and come on Monday, we discuss in detail. Thank you.
And this medial border, inferomedial border, it is also divided. Anterior part is known as medial orbital border and posterior part is known as medial occipital border. Okay, understood? Now we have separated, and this is a temporal pole. You can say this is lateral view of brain. We have separated the frontal lobe from the temporal lobe, and in the deeper plane lies the insula or insular lobe, insular cortex, also known as island of Rayleigh. Okay, this can sometimes asked in your viva. Another, which lobe is present? Fifth lobe is, name the fifth lobe, or island of Rayleigh can also come as your short note. Okay, and it is surrounded by this circular sulcus, and on the superior surface of this superior temporal gyri, there are 4 gyri are present, 2 small gyri, 2 long gyri. Small gyri are known as gyra brevia, long gyri are the gyra longa. Okay, so these are the, this is about insula. Now we are dividing the brain into 4 main lobes. Okay, by these sulcuses, important sulcuses. First is central sulcus, here is central sulcus, this is lateral sulcus and its posterior rami, and extend it posteriorly first imaginary line. And here from the preoccipital notch to parieto-occipital sulcus, this is the first imaginary line, here is second imaginary line. So all these lines together dividing the brain into 4 main lobes: frontal lobe, parietal lobe, temporal lobe, occipital lobe. Okay, understood? So in this view you can see all the 3 lobes, poles, sorry, occipital pole, frontal pole and temporal pole. Okay. Now we will discuss the main sulci and gyri on the superolateral surface. Okay. So first main sulci is the central sulcus. Central sulcus starts from the superomedial border 1 centimeter behind the midpoint between the frontal and occipital pole. Okay, from here it is starting, extending downward and forward and meeting this lateral sulcus. Okay, lateral sulcus, it has 3 rami: anterior horizontal, anterior ascending and posterior ramus. Okay, this anterior, okay, now we go from here, central sulcus, and parallel to it, anterior to the central sulcus is precentral sulcus, posterior to the central sulcus is postcentral sulcus. So in between central sulcus and precentral sulcus, here will be the precentral gyrus, which is the motor cortex. Okay, area 4 of Brodmann. And postcentral gyri lying between the central sulcus and postcentral sulcus, this is primary sensory area, Brodmann area 312. Okay. Now in the frontal lobe, 2 sulci are dividing the frontal lobe into 3 gyri from above downward. Superior and inferior frontal sulcus dividing the frontal lobe into superior, middle and inferior frontal gyri. Now this inferior frontal gyri, this is again divided by this rami of the lateral sulcus into 3 parts. This is anterior horizontal, this is anterior ascending. These are dividing the inferior frontal gyri into 3 parts: pars anterior, pars triangularis and pars opercularis. A, B and C representing pars orbitalis, triangularis and opercularis. Operculum means a lid. Okay, it is covering the insular cortex. This is frontal operculum and below there is temporal operculum. Okay. They are together covering the insular cortex from the surface. Now we go to the parietal lobe. Below this posterior to this postcentral sulcus, the rest of the area, there is only one sulcus, intraparietal sulcus, which is dividing the parietal lobe in the upper part superior parietal lobule, in the lower part inferior parietal lobule. Okay. Now we go to the temporal lobe. Here also two sulci are seen, superior temporal sulcus, inferior temporal sulcus, dividing the temporal lobe into three gyri, superior, middle and inferior temporal gyri. Okay, understood? Now this upturned end of the posterior ramus of lateral sulcus, around which one gyri is present, that is known as supramarginal gyri. This is area 39. Okay, supramarginal gyri is area 39. Next, around the upturned end of this superior temporal sulcus, this area is known as angular gyrus. This is area 40. Okay, these are important association areas 39 and 40. And these parts, this supramarginal gyri and angular gyri, these are located within inferior parietal lobule. Okay, understood? Next we go to the occipital lobe. Here there is extension of the postcalcarine sulcus. Around it, this U-shaped sulcus is the lunate sulcus, and here is lateral occipital sulcus, and here will be the transverse occipital sulcus. So these are the main sulci and gyri in the superolateral surface. You must know this central sulcus, precentral, postcentral sulcus, lateral sulcus is the three rami, you must know. Okay. Next we go to the medial surface. How will you identify it is medial surface? You can see the section of corpus luteum. Okay. What are the four parts of the corpus luteum? From anterior to posterior, there will be rostrum, genu, then body and splenium. Okay. So these are the four parts of the corpus luteum. Now see the important sulci and gyri of this area. Just above the corpus callosum, there is callosum sulcus. Okay. Parallel to the corpus callosum, above is the cingulate sulcus. Okay. In between the callosal sulcus and cingulate sulcus, here will be the cingulate gyri. Now here there is extension of the central sulcus here in this part, and around which anteriorly extension of the primary motor area, posteriorly extension of the primary sensory area. These two parts are together forming the paracentral lobule. Very, very important. Around the extension of the central sulcus on the medial surface, the area of brain is known as paracentral lobule. Very frequently asked in your viva. This is area for maturation and defecation. Okay. Now anteriorly, this part is forming the medial frontal gyri. Frontal gyri. Okay. Now posteriorly, here is one sulcus just above the splenium, that is known as suprasplenial sulcus. Okay. And this part is known as precuneus. Now this sulcus on the medial surface separating the parietal lobe from the occipital lobe, this is known as parieto-occipital sulcus, and this is known as calcarine sulcus, and its posterior extension is known as postcalcarine sulcus. So the part in between the parieto-occipital sulcus and the calcarine sulcus, this triangular part is known as the cuneus. Okay, understood? Next, we go to this temporal lobe. In this part, it is also divided by two sulci. One is collateral sulcus, another is occipito-temporal sulcus, dividing into three gyri. Parahippocampal gyri, the medial to this collateral sulcus lies this parahippocampal gyri. Then here is medial occipitotemporal gyri. Here is lateral occipitotemporal gyri. Okay, and anterior extension of this collateral sulcus. Here small sulcus is present. This is known as rhinal sulcus. Rhinal means related to nose. So olfactory area is located here. This anterior projecting part of this parahippocampal gyri, this is known as uncus. This first is very important. You must know the uncus. You have to show it in your viscera. Okay, so uncus, here lies the olfactory area, area 28. Okay, and in front of this rostrum, here lies the paraterminal gyrus. These are very small gyri, and another is parolfactory gyri. Next we go to the inferior surface. Inferior surface is divided by this lateral sulcus into posterior part is known as the tentorial surface, anterior part is orbital surface. Orbital surface, over the orbital surface, just parallel to the medial border, one straight sulcus is present. That is known as olfactory sulcus. Okay, why this name is given? Olfactory bulb, olfactory tract are lying along this sulcus. Okay, that's why olfactory sulcus. And one straight gyri is present just medial to this olfactory sulcus, that is known as gyrus recti. Okay, and rest of the part of the orbital surface, here one H-shaped sulcus is present, that is known as orbital sulcus, and it is dividing the orbital surface into 4 gyri: anterior, posterior, medial and lateral orbital gyri. Okay, understood? Next we go to the tentorial surface. Here you can see the extension of calcarine sulcus, postcalcarine sulcus, lunate sulcus, and these are the collateral sulcus, rhinal sulcus, and here is the occipitotemporal sulcus. And the 3 gyri, parahippocampal gyri, anterior extension is uncus, here is the medial and here is lateral occipitotemporal gyri, extending into occipital lobe and temporal lobe. That's why the names are given. Okay, so these are the important sulci and gyri of the inferior surface of the brain. Understood? So now we will discuss, go to the Brodmann's area. Okay, functional areas of the brain. So cerebral cortex is demarcated into large number of areas which differ from each other in structure as well as in function. Brodmann in 2009 had divided the cerebral cortex into 47 such areas and indicated each of them by a number. Okay, so these are the different Brodmann's area. On the superolateral view, you can see this is central sulcus. Precentral gyri is area 4, that is primary motor area. Postcentral gyri is primary sensory area. This is 312. Then here is the premotor area, area 6. Here is the frontal eye field, area 8, and prefrontal area is 9 to 12. Okay, 9, 10, 11 and 12. Next, in the pars triangularis, area 45, and pars opercularis is area 44. These two are together forming the motor speech area of Broca. Very important. Motor speech area of Broca. Next, posteriorly in the parietal lobe, you can see area 5 and 7. These are secondary sensory area and or sensory association areas. Okay. Now, around the upturned end of this posterior ramus of lateral sulcus, here is area 40 in the supramarginal gyri. Now, upturned end of this superior temporal sulcus, there is area 39. This is known as sensory speech area or Oendix area, that is mainly area 39. And in the posterior part of this superior temporal gyri, here is lying area 22. Mainly this area 22 is known as Wernicke's area, and these 3 areas are together forming the sensory speech area. Area 22 it is coordinating with sound, auditory impulse, 39 is getting the visual impulse, and 40 is getting different sensory impulse, touch, pain, temperature, pressure, like this. So these three are together coordinating and they are together forming the sensory speech area. Understood? And primary auditory area is area 41 and 42. They are lying on the superior temporal gyri, mainly on the superior surface of the superior temporal gyri. Here is 41 and around it will be the 42. Okay. So they are mainly, you have seen on this during the insular cortex, gyra longa and gyra brevia, those parts are the auditory area, primary auditory area. Okay, on the superior surface of the superior temporal gyri. Location is important. Now in the occipital region, around the calcarine sulcus, here is area 17 or striate area or primary visual area. Next, around 17, there is area 18, that is known as parastriate area. Okay, that is visual association area, and around 18 will lie the area 19, that is higher visual association area or peristriate area. Okay, understood? So these areas are very, very important. Motor speech area of Broca, sensory speech area, that is Wernicke's area, containing area 22 mainly, also 39 and 40. This primary motor area, primary sensory area, and here this visual areas, area 17, 18, 19. Okay, these are very, very important functional areas. Next we go to the medial surface. On the medial surface you can see uncus and parahippocampal gyrus. Here lies the primary olfactory area. Okay, here lies within the cingulate gyri 23 and 24, and in the paracentral lobule forming part of the motor area, part of the sensory area is lying. They are together forming the paracentral lobule, center for micturition and defecation. Here is the premotor area, extension of area 6, and here is 8, 9, 10, 11, also 12 is present. Okay, prefrontal gyrus. Here on this medial surface you can see the three visual areas. Around the calcarine sulcus there is area 17 striate area, 18 is parastriate, 19 is peristriate. Okay, primary visual area, visual association area, higher visual association area. Okay, so these are important. So three main types of functional areas are there: motor area, sensory area, and association areas. Primary, now we will discuss the motor areas. First is primary motor area, that is area 4. It is located in precentral gyrus in superolateral surface, and motor homunculus is present over here. Okay, this is representation of different areas. How the motor function is more developed, that area is more enlarged in the cortex. So on the medial surface lying the foot and lower limb, then hip, trunk, then on the superolateral surface from above downward you can see representation of the hip, then trunk. Okay, then there is the arm, forearm, then hand. Hand area is larger because very precise movements occurs in the hand. Face also precise movements occur, different facial expression, movement of tongue or during speech. Here is the tongue and larynx. So these are the representation of sensory areas in the sensory cortex. Okay, that is known as, motor areas in motor cortex, that is known as motor homunculus, and they control the voluntary movements of the contralateral side. If paralysis occurs in the left hemisphere, right side of the body will be paralyzed. Okay, like this, vice versa. Now effect of lesion of the primary motor area: contralateral flaccid paralysis initially, decreased deep tendon reflex, Babinski sign positive, and moderate recovery of function occurs because it is upper motor neuron type of paralysis. Okay, in lower motor neuron type of paralysis there will be complete paralysis, no recovery. Next we go to premotor area, that is area 6 and 8. Here this is primary motor area, here is 6 and 8. Area 6 responsible for skillful acts, programming of movements. Upper part of area 6 is the writing area. Area 8 is also known as frontal eye field, regulates the voluntary conjugate movements. Next we go to supplementary motor area, continuation of area 6 on the medial surface. Here this is supplementary motor area. It controls the complex movements, elicit and urge to move. Lesions of this area will lead to akinesia, loss of movement, and upper motor neuron type of paralysis. In this case, there will be conjoint lesion of primary motor, premotor and supplementary motor areas. All these areas are paralyzed. Okay. Now, prefrontal area, that is area 9 to 12, 9, 10, 11 and 12. In front of premotor area, it is like premotor area was 6 and 8. They are highly developed in human, prefrontal area. These functions are very frequently asked in your viva of prefrontal area. Mainly all complicated tasks, higher thinking activities, all those are performed in this frontal lobe. Okay. So it regulates the depth of feeling, abstract thinking, mature judgment, foresight and tactfulness. This is the silent area of the brain. Okay. Next we go to the sensory areas. So we have covered the motor areas. Now going to the sensory areas. There are primary sensory area, secondary sensory area only for somatosensory senses, sensory association area and higher association areas. Primary sensory area, these are again of three types: somatosensory area, visual area, auditory area. Primary somatosensory area, it is located in the postcentral gyrus on the superolateral surface. Here this is area 312, that is primary sensory area or primary somatosensory area. What is the function? It localizes, analyzes, discriminates different modalities of cutaneous sensations like touch, pressure, position, vibration senses. Sensory homunculus are present here. Sensations are somatotopically arranged. Here also you can see on the medial surface there is foot and lower limb, then there is on the superolateral surface trunk, head and neck, then arm, forearm, then hand, face, like this. Okay. So this is the representation of sensory homunculus in the sensory cortex. Lower part of postcentral gyri is the taste center, that is area 43. Okay. Here postcentral gyrus lower part in this region will be area 43, that is taste area. Next we go to the primary somatosensory area functions. One important part is paracentral lobule, that is area for maturation and defecation, lying on the medial surface, receives senses of distension of bladder and rectum. Okay. Areas are 4 or 3, 1, 2. দুটোই থাকবে. Next we go to secondary somatosensory area or supplementary sensory area. Upper lip of the posterior ramus of lateral sulcus, lower part of pre and post central gyri. This is the location of secondary sensory area. Functions: it receives the pain sensation and other modalities of cutaneous sensations. Okay. Next, sensory association area, that is area 5 and 7. Here area 5 and 7 lying in the parietal lobule, superior parietal lobule mainly, behind the postcentral gyri, superior parietal lobule location, area 5 and 7, somatosensory association area. Next, higher association area for somatosensory senses. Somatosensory sense means touch, pressure, vibration, like this. Okay. So higher association areas are present in the supramarginal gyrus, area 40, and richly connected with area 5 and 7. Lesions of this area 40 will lead to tactile agnosia or astereognosis. Okay, astereognosis means you cannot understand anything by touching the object if you close your eyes. Okay, and tactile aphasia. Now olfactory area, Broadman's silence কি কথা বলছিস এত আগে শুনে নে তারপর নিজেরা ডিসকাস করিস olfactory area is area 28 is located in the anterior part of parahippocampal gyri and uncus here primary olfactory area 28 uncus and anterior part of parahippocampal gyri. Next is visual senses, primary visual area 17 you have seen, visual association area 18 and 19, higher visual association area is area 39 here, okay, in the angular gyri. Now visual association area 18 and 19, parastite area is area 18 also known as visual area 2 and peristite area is area 19 also known as visual area 3. What are the functions? These are known as occipital eye field area 18 and 19 and mediate conjugate movements involuntarily they helps in conjugate movement of the eye. Conjugate movement means both eyeball will move together. Okay. Next is auditory area. Auditory senses, primary auditory area is area 49. Auditory association area is area 42 and higher auditory association area is area 22. Here you can see 41, 42, 22. Okay. In the superior temporal gyri. Psychical cortex, anterior part of temporal lobe is known as temporal pole. Here, temporal pole. This is known as psychical cortex. What is the function? It helps in memory, present and past memory, dreams, hallucinations, auditory and visual hallucinations occur in this area. There is a circuit formation here. Okay, from hippocampus, parahippocampus, amygdaloid body, then this on the medial surface, cingulate gyrus, all these are together forming a circle, C-shaped circuit-like structure. Those helps in memory. Okay. Now speech. Speech is very, very important. It is performed by left cerebral hemisphere regardless of the hand preference. Hence, left hemisphere is dominant or talking brain and right hemisphere is non-dominant or mute brain. Now, dominant hemisphere has 4 speech centers, 3 sensory and 1 motor speech center. Sensory speech areas are these areas. Area 22 is higher auditory association area. This is known as Wernicke's area. Area 39 higher visual association area and area 42 higher association area for somatosensory senses. Okay. So, amra kokhon we generally talk after reading or after auditory hearing. Okay. So, all those senses are together associated in these higher areas. After that we can talk after understanding all those things. Visual or okay. So, all these things, these areas are together forming the sensory speech area. Next is motor speech area is located in area 44 and 45, pars triangularis, pars opercularis, Broca's area, very, very important. So, speech centers, this motor and sensory may be transferred to the right hemisphere in child up to 6 to 8 years of age in case of cerebral injury. Okay. Generally, it lies in the left hemisphere in both right and left-handed person. But in case of child, if injury occurs on the left hemisphere, the speech center can be transferred to the right hemisphere. Okay. Now, this area 22, 39, 40, they receives input from hearing, vision, touch and proprioception. 40 te chilo touch and proprioception, 39 a vision, 22 te hearing. Okay. All these senses are associated here. And so, information processed in area 22 posterior part and projected to Broca's area through arcuate fasciculus. Arcuate fasciculus is connecting sensory and motor speech area. See here, the green area is the sensory speech area, 22, 39, 40, and this is motor speech area, Broca's area, 42, 44 and 45. These two are connected by arcuate fasciculus. কোথায় গেছলি তুই? ছুটি হয়ে গেছে যা। Okay, so lessons of speech area, lessons involving speech area, speech center or dominant hemisphere, it will lead to aphasia. Two types of aphasia is present, sensory aphasia, motor aphasia. Sensory aphasia, sensory speech area is destroyed, sensory aphasia, patient cannot understand anything. Okay, motor speech area, if motor speech area is destroyed, there will be motor aphasia, patient cannot talk but patient can understand. Okay, and conduct aphasia, lesion of arcuate fasciculus, connection between sensory and motor area. There will be fluent, when a patient can talk and also patient can understand, but there is no correlation between understanding and talking. Okay, so there is fluent, patient is fluent, comprehension is also intact, and repetition of spoken language is extremely difficult in this conduct aphasia. So sensory speech area is located in the left dominant hemisphere, occupying the posterior part of superior temporal gyri of the temporal lobe, angular gyri area 39 and supramarginal area 40 of the inferior parietal lobule. And the Wernicke's area is concerned with the interpretation of language through visual and auditory input, and it is also an essential zone for constant availability of learned word patterns. The angular area or area 39 and supramarginal gyri are essential for process of learning. Okay, they are higher association areas, so learning is processed here. Now summarization, we have three types of areas mainly. Motor areas mainly in the precentral gyri, sensory area, postcentral gyri, psychical area at the temporal pole. Okay, motor areas are primary motor area, area 4 premotor area, 6 and 8, supplementary motor area or prefrontal area. Supplementary motor area extension of area 6 on the medial surface, our prefrontal area was 9 to 12. Okay, so these are the motor areas. Next is sensory area, primary sensory area, area 312, secondary sensory area only for somatosensory senses 5 and 7, sensory association area is say area 5 and 7 lower part, higher association area 40. Okay, now primary sensory areas are somatic area. Somatic area is area 312, visual area 17, 18 and 19, auditory area 41, 42 and 22 is higher auditory association. Okay, so these areas are important, all those you have to see. Primary motor area, first the central sulcus, primary motor area 4, primary sensory area 312, motor speech area, Broca's area 44, 45 in the pars triangularis, opercularis, and sensory speech area 39, 40, 39 is supramarginal gyri, 40 is angular gyri. Okay, and after that the occipital pole, visual area 17, 18 and 19, auditory cortex 41 and 42 and area 22, part of 22 in the superior temporal gyri. Around the medial surface you can see the olfactory area is area 28. Okay, and paracentral lobule you have to see, and also the taste area 43, lower part of the postcentral gyri. Okay, so these areas are very important, you have to see the important sulci and gyri. Okay, now come to the table. That's all, thank you.